Para continuar con nuestro programa que tenemos definido el día de hoy, continuaremos con la presentación del doctor Daniel Tapia, y para ello me permitiré leer una breve reseña para conocer a nuestro próximo conferencista. El doctor Daniel Tapia es científico senior de Ciencias de la Expresión de Proteínas en Regeneron Pharmaceuticals. Él es orgullosamente mexicano, propiamente de Morelia, México. El doctor Daniel Tapia se unió a Regeneron hace 2 años como científico en el Departamento de Ciencias para la Expresión de Proteínas, PES por sus siglas en inglés. Como científico senior, el doctor Tapia se enfoca en el descubrimiento de anticuerpos generados en diversas enfermedades, colaborando estrechamente con equipos multifuncionales para optimizar nuevas estrategias que mejoren la eficiencia en la producción de anticuerpos. También colabora con diferentes grupos dentro de Regeneron para implementar tecnologías terapéuticas adicionales. Antes de unirse a Regeneron, el doctor Tapia fue investigador del Region Institute en MGH, MIT y Harvard, donde trabajó en el desarrollo de nuevas estrategias terapéuticas dirigidas a vías inflamatorias en el contexto de las infecciones por virus de la influenza. El doctor Tapia tiene un doctorado en microbiología e inmunología de la Universidad de Texas Medical Branch, así como un título de biología molecular y celular de la Universidad de Texas A&M. Les pido un aplauso, por favor, para el doctor Daniel Tapia.
Bueno, muchas gracias al director Paul, al doctor Javier Sánchez por la invitación. No es mi primera vez en Cicate, yo creo que ya me debería dar un título ya de estar aquí. La manera de que yo siempre presento es un poco informal. Me encanta hacer un énfasis aquí a los estudiantes, a todo el aprendiz, que yo pues somos la mayoría. Particularmente, nunca saben por quién van a estar sentados al lado. La doctora Camila y yo nos graduamos exactamente el mismo año y trabajamos a 10 minutos de cada año. Ella trabaja en Pfizer, nos divide un río y hemos tenido tantas conversaciones de cuál es la mejor manera de identificar un blanco, ¿no? Bueno, empezamos los dos en el desarrollo de vacunas, pero muy pronto me di cuenta que a veces la espícula del virus, a veces es una proteína estructural o no estructural de alguna manera. Pero trata de cuenta de que hay veces no sabes cuál es el blanco. En muchas de las enfermedades, especialmente algunas de las más complejas, ¿cómo encuentras un blanco terapéutico? ¿Cómo lo validas y cómo lo empiezas a desarrollar análisis para poderte asegurar que ese blanco sea específico y vaya a ser efectivo en contra de la enfermedad? Entonces, bueno, para esto voy a dar mi plática también en inglés, pero sí quiero aquí que cualquier persona que tenga alguna duda, alguna pregunta, me pare y podemos responder cualquier pregunta que tenga. Bueno, el título de mi plática hoy va a ser Decoding the IL-6 dual signaling, targeting the right balance against viral infections at the respiratory interface. Y similar, the following slides were conducted at the Reagan Institute, and the views and opinions expressed in this presentation are solely my own and don't represent either Regeneron or MGH, MIT, or Harvard. Entonces, podemos continuar. Ahora, voy a dividir la plática en 2 secciones. In the first part of my talk, I'm going to introduce a little bit about the concept of inflammation and IL-6 and how striking the right balance between pro-inflammatory and suppressive responses can lead to either survival or perhaps succumbing to infection. So I'll talk a little bit about the differences between both signaling modalities of the IL-6 cytokine. And then I also talk a little bit about Regeneron, particularly being a biotechnology hub, and today one of the leading companies in monoclonal antibody therapies, but also genetic medicines, and as well as delivery of different modalities, particularly in the field of oncology and genetic diseases. So, perfect. I'll start talking a little bit about the coordinated balance of IL-6 trans signaling as a potential antiviral target against influenza virus infection. Perfect. So we know that IL-6 is a very crucial cytokine involved in inflammation and really responding to any immunological insult that the body receives. We know that IL-6 is very important, not only in inducing the initial innate responses, but also in a plethora of different mechanisms
including also the development of the adaptive response. Particularly, IL-6 is an inducer of B cell to plasma cell differentiation, which allowed for the secretion of antibodies. So we know that IL-6 plays a very, very important role in responding to any immunological insult, whether it's a viral infection, bacterial infection, or just any kind of immunological intolerance of the system. However, we know that if IL-6 levels are not correct, or there's a dysregulation in IL-6 responses, this can also lead to the death itself. So how do we know what the right mechanism is? How do we balance at the right amount the protective IL-6 responses and prevent against pathogenic IL-6 responses? So we know that IL-6 is a crucial player in inflammation, in the development of cytokine storm, which leads to systemic organ failure and even death. IL-6 has a classic signaling modality, which involves the binding of IL-6 to the membrane form of the receptor known as the IL-6 receptor. And in order for the signaling event to occur, it actually has to bring in a co-receptor known as GP130. This is known as a classic signaling response, and traditionally it was thought to be the protective response. And there was a way to differentiate between a protective IL-6 signaling and a pathogenic IL-6 signaling. However, go next. The IL-6 response also has an event known as trans-signaling. Classic signaling is only restricted to very particular immune cell subtypes and hepatocytes, but all of our cells in our body are responsive to IL-6. How does that happen? IL-6 can bind to a soluble form of the IL-6 receptor, known as the soluble IL-6 receptor, and bind in the circulation, so in sera or in the blood, and any cell in our blood type, any cell in our organism, it expresses GP130. So GP130 is expressed in all cell types, and that's why they can be responsive to IL-6 signaling. However, to complicate matters even worse, GP130 also has a soluble form of the receptor, which is known as soluble GP130, and it's known to be the brake of trans signaling. So trans signaling has a natural brake in the blood system. So imagine this system being a very important system, which you need to know where your IL-6 levels are, where your soluble IL-6 levels are, but also your GP130, your soluble form of the receptor. All of these different components will make a buffer mechanism in the blood that will basically control when a response is pathogenic versus when it will be a protective response. So keep that in mind. In development, there's one monoclonal antibody that has been developed to counteract the response of IL-6, and that's known as tocilizumab. Yeah. Perfect. So tocilizumab is the only FDA-approved monoclonal antibody against IL-6. However, it only binds IL-6 to IL-6 receptor. And if you remember from my previous talk, you'll know that IL-6 and IL-6 receptor are both found in classic and trans signaling. So there is no way to differentiate between the classic and the trans signaling response from this monoclonal antibody therapy. So initially in my postdoc at the Reagan Institute, we took on the job to try to understand more of these components in the blood and what makes them pathogenic and what makes them protective. So next. Perfect. Foundational work was done by a previous postdoc in the lab at the Reagan Institute. And one of the crucial questions that remained were, what is the source of the soluble IL-6 receptor? If soluble IL-6 is what controls this buffer together with the soluble GP130, what is the main producer? Through different transgenics in mouse knockouts, we found out that conventional dendritic cells are actually the main producer of soluble IL-6 receptor. That was very crucial for us to understand what mechanism is happening that leads to IL-6 being protective versus pathogenic. So if we go next. If you remember from what I mentioned earlier, is all of the different components in the IL-6 signaling response have very defined levels in the bloodstream. IL-6 can be of a wide range depending on whatever the immune system is encountering. If it's a viral infection, some levels go up initially right after infection, and then eventually they come down. So the IL-6, however, the IL-6 component can range in the picogram to milliliter in the blood. However, and the soluble IL-6 receptor has a much more defined range of concentrations in the bloodstream. What was really not known was happening to GP130. What are the levels of GP130 in the blood? And that's a very important question that not a lot of people had asked. They always ask, how much IL-6 do I have? Is it good? Is it bad? Can I measure a patient and see, is the IL-6 level good or is it bad? But no one had asked the question, what happens to GP130? Not only that, the affinity for each component it's actually more striking. The affinity from IL-6 to IL-6 receptor is in the nanomolar range, so good, but the affinity of the soluble IL-6 receptor to the natural inhibitor is a lot stronger. It's in the picomolar range. That says that that system has to be extremely tightly regulated in order for you to prevent IL-6 signaling response. So if we go next. So one of the initial questions we just asked is, well, what happens during a natural infection? What happens to a mouse during a sublethal infection? Perfect. So we infected mice with influenza H1N1 and the sublethal dose, so just to get an understanding of what happens to the response when the virus is not going to kill the mouse. So what happens is in a wild-type mouse, the IL-6 levels, as expected, they go up and they come down. The mouse is fine. The mouse was protected against infection. What happens when you don't have soluble IL-6 receptor? Those levels of IL-6 go through the roof. And some mice obviously will succumb to the infection, but not because they will succumb to the infection because of influenza, right? They will succumb to the infection because the levels of IL-6 are significantly dysregulated when you don't have the soluble IL-6 system response. Okay, so if we go next. So then we ask the question, if you remember what I said, what happens to the concentration of the different components in this system? If we measure the soluble IL-6 receptor, we know that also those tend to have a bell curve response where they go up and they come down, and that's what will grab to the soluble IL-6 and out it will neutralize and prevent the signaling from going dysregulated and killing the mouse. What happens to the soluble GP130 inhibitor of trans-signaling? This is something that not a lot of people had asked the question. If we look at during infection, right after you infect the mouse with a sublethal dose of influenza H1N1, we notice a decrease in soluble GP130. What is that saying? That the mouse is trying to push for more trans signaling and decrease the natural inhibitor to be able to be protected. So this was the first indication that we had that trans signaling was the protective response, and classic signaling, the soluble IL-6, was the pathogenic response. So if we go next. So now we know that the natural soluble GP130 that blocks classic signaling is downregulated in a sublethal mouse model of infection of influenza H1N1, suggesting that if you naturally were to decrease the levels of soluble GP130, you would potentially drive better and more protective IL-6 signaling response. So now, can we study more, and what can we understand in terms of how do you differentiate a monoclonal antibody against GP130 and perhaps lower the levels of soluble GP130 so you have increased protective responses from IL-6? What are the indications in patients? That's just another slide. We basically, this is where we show that this system was in response specifically in the TLR7 sensing of double-stranded RNA from influenza, and we knew that this system only occurred when you actually had active sensing of influenza virus infection in the presence of TLR7. So if we go next. Just once again, summarizing the results, that this was the first indication in a mouse model where trans signaling was perhaps involved in a protective response against influenza virus infection. Going up next. And in vivo, we took a mouse model of overexpressing the natural inhibitor, so the soluble GP130. Overexpression of this, what would we anticipate? If we had overexpression of the inhibitor, we would be blocking trans signaling, and we would expect a higher frequency of death in mice in response to influenza virus infection. So if we go here, that's exactly what we saw. We noticed that if you completely knock out IL-6, the mouse cannot respond to an influenza, and they will die as a response to infection. However, when you overexpress the inhibitor of trans signaling, you have an equal frequency of death, almost equivalent to the complete absence of IL-6 signaling. So this was the first in vivo response. We asked ourselves, what happens in COVID-19 patients? What happens when we stratify and we take patients all the way from healthy to people that succumbed to the infection. So we collaborated with a group that had done a cohort of study of stratified patients in response to COVID, and we looked at those different levels of the different components of the buffer system. So then we looked in patients that were more predisposed to succumbing to the infection against COVID-19. We noticed obviously higher IL-6 levels. This is the field. This is what we know. IL-6 levels can be very pathogenic if dysregulated. No difference in soluble IL-6 receptors, so no difference in the buffer, but you still have uncontrolled levels of IL-6. But what we noticed was a significant upregulation in the presence of soluble GP130. So this was saying that patients that died as a response to the infection probably had lower propensity of trans-signaling response in response to COVID-19. So then, this is still ongoing studies at the Reagan Institute right now, which is trying to determine how can you now that we've identified the target, now that we have the target against where we can actively target the responses against soluble GP130 to develop a good monoclonal antibody therapy that perhaps neutralizes better or perhaps acts better than tocilizumab, which only binds to IL-6 and IL-6 receptor. We begin to ask the question is how can we generate a response that's specific for trans signaling but not classic signaling? So this is the ongoing work. We immunized several mice. And sorted B cells. This is kind of going back into how monoclonal antibodies are discovered. We immunized mice with either the mouse form or the human form of soluble GP130 in different formats. So we wanted to make sure that we were very specific to a conserved epitope within the soluble GP130 binding motif. And then we sorted for specifically the stalk, so what anchors the soluble GP130 onto the membrane. We took B cells from immunized animals. We sequenced their B cell receptor repertoire, so we could actually clone those variable domains into different expression vectors. We would then transfect our eukaryotic expression systems, and then once we transfect with our known variable domains, then we would just purify a monoclonal antibody therapy. And we could see a head-to-head comparison. What monoclonal antibody therapy would work better? Is it tocilizumab that completely ablates IL-6 signaling response? Or would it be something that's perhaps more specific to a protective mechanism and not a pathogenic mechanism of IL-6 signaling? So just a short conclusion from the studies that we conducted in understanding systemic IL-6 responses. So in mice, systemic IL-6 concentration is regulated by a blood buffer system that actively shifts to enhance IL-6 trans signaling following a sublethal infection by influenza virus. In mice, IL-6 trans signaling is required to survive lethal infection by influenza virus, and in humans, buffer shifting to enhance IL-6 trans signaling appears to be inversely correlated with disease severity. So this was the first indication that trans signaling, not classic signaling, was the protective modality of IL-6 signaling responses. Perfect. This is work that I was able to carry out at the Reagan Institute in Boston. The Reagan Institute is a world-leading institute for the discovery of antibodies against influenza, against HIV, and also vaccines against what are known as vaccine-resistant pathogens, because even if you develop a vaccine, they're so complex in their mechanism that there may not just be one vaccine that protects against all of the different forms of these pathogens. So I'll shift gears to talk a little bit about Regeneron and why I decided to, leave academia and go work at Regeneron, which is a leading biotechnology company. And the reason why I really made a decision to go with Regeneron was because of their potential scientific breakthroughs that they've developed. And they really were one of the first, if not the first company to develop a fully humanized mouse with a B-cell receptor repertoire as a human. So basically, if you immunize a mouse with anything, it will develop an antibody response as a human would. These are gigantic DNA sequences that are coding for the individual variable domains in an antibody. So Regeneron was a leading pioneer. And one of the things that you'll see throughout my talk about Regeneron is Regeneron is really a company that is founded on developing a strong platform for developing therapeutics. So not buying different smaller companies that develop the therapeutics, but building a pipeline where you understand everything from the discovery, but also from the target identification. So how do you know what isoform of a protein is the one that's dysregulated in a patient? So everything from the target discovery to validation to the actual implementation and the development of a biological therapeutic like an antibody. So Regeneron is a leading technology hub that has developed many kinds of different platforms, including the very first bispecific antibody. And so now, if we talk into the context of antibodies, a single monoclonal antibody, we know it's not going to be effective against an oncological target, such as single cell carcinoma, but we know that bispecific responses today and bispecific molecules are the leading candidates for oncological targets. So we go next. As of today, Regeneron has 12 FDA-approved medicines, and we do pride ourselves in saying that of all the different therapeutics, 100% of them have been completely done at Regeneron. Everything from the target discovery to the validation, to the manufacturing, to the approval, everything is being done and pioneered at Regeneron. We have in the pipeline, we have over 35 new molecules. And we do publish significantly. It's still very much, if you think about it, it's almost sort of a publishing hub with new discoveries happening every day. I don't know if you have heard about the genetic study that was being done here in Mexico, which was looking at the genetic diversity of the Mexican population here. There's actually work that was actually being done at Regeneron as well in collaboration with Oxford and different groups throughout the world. We go next. As I mentioned before, we know that Regeneron really takes sort of what big pharma mindset has and completely turns it on its head and develop their own. Everything from, for example, the decision-making process of a company. Conventionally, it's a lot of it's commercial. Is it feasible? Is it cheap? Is it the best? Is it the best decision that we make? At Regeneron, everything from every single decision is still very much a science-driven decision. What does the result say? What does it say? Where is it pointing you to? As opposed to different companies, like I said, that focus sometimes on one drug or one disease or a very particular therapeutic area, Regeneron tries to cover everything. And that really allows them to be able to control and to learn from everything from the identification of a target to the validation to this and the manufacturing. As I mentioned before, we have 12 approved medicines in the US and in other countries. Particularly, I'll talk a little bit about the oncological targets that are coming in the pipeline. But everything at is being developed from the very early principle basic science approach down to the manufacturing and large-scale production. This is just the current pipeline that we have in different phases. These are all different targets and also now the different delivery mechanisms. So oftentimes, monoclonal antibodies are just given straight into as a single or multiple injections. However, there are many diseases where you need to specifically get it somewhere. Is it in the brain? Is it specifically into a very specific tissue? So now there's a lot of developments being done in adeno-associated viral delivery of these biological therapeutics, but also genetic delivery as well. So that's why there's a couple of conjugates and monoclonal antibody conjugates to siRNA for downregulated responses against a very specific mRNA molecule. As I mentioned before, Regeneron really does sort of pride themselves in the technology pipeline that they've built. And everything at Regeneron is called the Velocity. Somebody really liked dinosaurs, and so everything is labeled as a Velocity. So the very first mouse model that was developed to create a B-cell repertoire, so create monoclonal antibody therapies, was actually, if we go next, is actually the Velocity mouse. So what allows Regeneron to be a leading monoclonal antibody company is really being able to control all of the different areas in the identification and production of antibodies, down from what I mentioned, the target discovery and validation, the understanding what kind of molecule. Because once you understand what target is, what kind, how am I going to deliver it? What's the best mode of delivery? All of that, the manufacturing. It's not good enough to get a really good drug that you cannot produce, and that you cannot produce in sufficient amount. So also you have to be able to control how you're going to produce it. How are you going to now implement it? What if this is going to go straight into the eye? How is somebody going to apply something straight into the eye? So these are all considerations that we have to understand and innovate. So Regeneron has now they developed this, it's a syringe that as soon as it injects, the needle will retract. So that anybody that wants to do it at home can actually do it, and the needle would automatically retract. So everything, innovation happens at all stages of Regeneron. We go next. And as I mentioned before, what really allowed Regeneron to pioneer monoclonal antibody discovery is how fast we could develop a mouse model that was fully humanized for their B cells to respond to any kind of antigen in the velocity gene. So this is a pioneering technology that allows for genetic modification in the mouse genome. And this is also very important because for many targets, you actually have to immunize a mouse. So if the target is not human, for example, if it's CD40, if the molecule is CD40, I'm just throwing an example, and you have to humanize it, you have to have that mouse model in order to validate your anti-CD40 antibody. So this is very slow, but by creating these technologies and by being able to control everything about them, you're able to learn from everything and implement and always innovate in your own in your own new niche. So next, this is just some of the groups that take part in the in the clinical pipeline, in the preclinical and clinical pipeline of Regeneron. So at the very initial discovery phase, remember what I said, there's usually some sort of a group that gets together and says, what disease should we focus on? What molecules are out there? What is known in the literature? But not only that is, how can we here identify our own validated targets? And then how do we make it? So once you have your target of choice, this is actually where I come in, protein expression sciences. In protein expression sciences, we will actually discover the antibodies, test them, do preclinical tests, and then also develop the cell lines, the cell lines that will later express your antibodies that you want to produce. This is just in a very, very small snapshot of what protein expression sciences does for Regeneron. The velocity gene, like I mentioned before, they come in developing the mouse model first for the disease that you want to study and the disease you want to develop a target for. And then we now have to identify and create those antibodies. So that's what BST stands for, B cell sorting technology. So that's where we discover the antibody. And then now the easier, the nicer, and the faster are actually cell lines. These are genetically characterized, fully genetically characterized cell lines for optimal high expression of antibodies. We are talking 7 to 12 grams per liter of cells. That's how incredible that those cell lines have to be. So now, Regeneron currently is pioneering a lot of the oncological approaches for monoclonal antibody therapeutics. There's 3 different major classes. There's the co-stimulatory, so the CD3 and CD28 co-stimulatory bispecific. Those are going to help you engage your T cells and activate your T cells. There's also the tumor-specific bispecifics. So the bispecifics will recognize a tumor-associated antigen. They will then also recognize your T cell, bring them in close proximity in order to induce T cell-mediated killing. But now also the anti-PD-1, the checkpoint inhibitors, those inhibitors are going to be very important to help you make sure that the T cell responses are active and that your specific bispecific antibody is good. So that is a pioneering technology in building antibodies that have now 2 different recognition motifs. One recognizes the T cell, one recognizes the cancer, brings them together, and has some very specific responses to develop. These are just some of the ones that we're currently testing. So you can see there's a wide range of different kinds of oncological targets. There's everything from melanoma to ovarian cancer to prostate cancer. And those really have different categories. So we have the CD3 bispecifics that will help you engage your T cells. Well, some of the ones that will help with the co-stimulation, so your anti-CD28, but also your checkpoint blockade inhibitors, so your anti-PD-1 that will help you maintain an active T cell response. And also, Regeneron was in the headlines during the COVID-19 pandemic. During the COVID-19 pandemic, they really also pioneered a antibody cocktail that's actually the very first implemented, even before the vaccine was out there, in a record time between 3 to 6 months to go from not knowing what your antigen will be to having a fully monoclonal antibody therapeutic in the pipeline is incredible. That's the one that Donald Trump said, the cocktail that he was given. Next. As I mentioned before, the Regeneron Genetics Center is a hub for understanding what the target will be. The Regeneron Genetics Center is sequence about 2.4 million exomes in individuals. That's an incredible number of genetic characterization in order to identify, for example, target variants and understanding genetic variation in a specific population. And this is done obviously in collaboration with over 130 research institutions. Out of these already, we have 30 novel therapeutics that have come directly out of sequencing exomes at Regeneron, and about 20 that have been genetically validated to be the target of choice for the specific therapeutic modality. Next. As I mentioned before, therapeutic monoclonal antibodies are now sort of the old son of Regeneron. There's now a lot of development being done in taking the idea of siRNA conjugation or siRNA delivery and siRNA therapeutics into the clinic. So we have a couple of clinical trials helping deliver siRNA to downregulate specific genes. We also have a collaboration with Intellia, which is the leading genetic delivery of CRISPR-Cas9 to help deliver and edit whole genomes. And then obviously genetic delivery via AAV and also antibody delivery. As I mentioned before, Regeneron has a wide sort of footprint in the production itself of monoclonal antibodies. We have a huge facility in Rensselaer, Regeneron, that is about 1.8 million square feet of production lab. This is very obviously very large scale bioreactors that allow for the production of monoclonal antibody therapies. And we have a history of launching a product on the very day that it is FDA approved. And the reason why we can actually do that is because by the time the FDA has approved the drug, it's already being made. That's how quickly they can get it out into the clinic. Perfect. So this is this is a very nice slide summarizing all of the different technologies that Regeneron is taking on, using still very much the foundational work that was done in mouse genetics and taking on everything from understanding human genetics, human identification of target variants, and taking on and delivering all of these now modalities. So not only monoclonal antibody therapies, but genetic medicine and different modalities of delivery. And that's what brings me here today. I love education. I strongly believe that many of the reasons why there's not representation of underrepresented minorities, so people from Hispanic and Latin backgrounds, is because we don't know a science is there, or that's what people say, right? So Regeneron is now the official sponsor of ISEF, that is the largest international science and engineering competition for all ages. And they are now the official sponsor. It used to be IBM, but now Regeneron is now the official full sponsor of that. And Regeneron has educational programs for all of the educational areas, everything from high school, so everything from your still primary education to leading the international high school and graduate level engineering competitions, a summer research internship program that I'm hoping one day I can bring an individual from here in order to stay at Regeneron for about 3 to 4 months. And this allows them to sort of transition onto full-time employees. We also have the Regeneron co-op program that's about 6 months in length. And then now we even have a postdoc fellowship that's it's specific for industry. And it's about a 2 to 3 year program. These are actually two groups of elementary school students. Unas eran de Puebla y las otras del Estado de México y fueron a competir. Y pues imagínense, fui juez en esta competencia y pues es extremadamente orgulloso de ver cuando pasaron los equipos de las dos niñas a competir desde aquí. Y bueno, con eso, ya, bueno.
Muchas gracias, Daniel, por compartirnos. Pues es el momento de las preguntas. Seguramente ya hay varias manos levantadas. Si me ayudan con el micrófono, empezamos. Aquí está una jovencita de... Ahí está el micrófono. Adelante con tu pregunta, por favor.
Hola, buenos días. Muchas gracias por la conferencia. Y bueno, mi pregunta es que si han estudiado el GP130 en el VIH.
GP130, bueno, la mayoría de los estudios que se han hecho son con estudios de una enfermedad aguda, que rápidamente te ayude a interrogar la respuesta inmediata, y una respuesta crónica específicamente mediado por citocinas de la inmunidad innata hace un poquito difícil. O sea, no conozco el estudio de ahorita, pero me imagino que sería una pregunta interesante de ver los efectos crónicos de una enfermedad como el VIH. Definitivamente, sí. ¿Excelente?
Gracias.
Sí.
Sí. Hay 3 manos levantadas. Ahorita llega el micrófono. Un segundo, por favor.
Hola, buenos días.
Hola.
Muy interesante tu plática. ¿Han medido ahora tanto GP130 como la interleucina 6 en pacientes infectados con las nuevas variantes? Me refiero a que ya no vemos la tormenta de citocinas, toda la respuesta inmune e inflamatoria ha variado. Entonces, ¿qué están viendo ahora?
Sí, no, bueno, ese trabajo lo hice cuando estaba haciendo mi postdoc. Es un proyecto que sigue y que no solamente ha dado muchos frutos, porque exactamente, o sea, cuando estamos hablando de una citocina que pues es de importancia clínica, no solamente los niveles, sino también las diferencias y paciente. Es una citocina que es tan variable entre paciente a paciente, entre género, mujer, hombre, son niveles extremadamente variables, pero sí sería algo muy, muy importante comparar las diferencias entre las diferentes variantes del SARS-CoV.
Gracias.
Hacia atrás también hay 2 manitas levantadas. Enseguida les llega el micrófono. Tienes mucho trabajo, Daniel.
Hola, doctor Daniel.
Sí. Ya.
Buenos días.
Hola, buenos días.
No me presenté hace rato, soy Randall de la UAM, de la Metropolitana, y tengo 2 preguntas. La primera es respecto a la parte de SPG 130, y comentó acerca de que está regulado la baja y de la parte de la expresión. Entonces, mi pregunta va un poco acerca de las técnicas de si se vio que está regulado a la baja, ¿qué técnicas más valdría la pena realizar? RNA-seq, ChIP-seq, y teniendo en cuenta esto, si valdría la pena hacer un análisis de expresión diferencial para evaluar qué genes están regulados y cómo se podrían atacar. Y mi segunda pregunta va más en función de qué opinión y perspectiva tiene usted acerca de la medicina personalizada y si cree que Regeneron puede atacar este mercado y cómo lo puede atacar.
Claro. Excelente pregunta. Sí, o sea, literal, la respuesta puede ser tan compleja como la vía de señalización del IL-6. Y sí, como se dio en cuenta, la presenté como la parte superficial de la vía de señalización, pero es en realidad abajo donde están las diferencias, pueden ser. Los niveles son extremadamente importantes porque son los que van a llevar a cabo la vía de señalización, pero exactamente, de hecho, muchos de los estudios que empezamos a hacer al final fue exactamente eso, es ver a nivel transcriptómica las diferencias de expresión, es particularmente obvio por siendo STAT3, que prende y apaga todo, exactamente de entender las diferencias de expresión en pacientes. Particularmente te pudiera decir no solamente eso, pero también a nivel celular, entender qué células están siendo reclutadas en respuesta al IL-6. Rápidamente puedo decir que es una respuesta, si se dieron cuenta, todos esos niveles son al nivel de respiratorio, por eso también el título de mi plática, que no recalqué tanto, pero en realidad el IL-6 es muy promiscuo. No es lo mismo que esté siendo en el pulmón a sistémico, o no es lo mismo que esté en el hígado a que esté en el... Entonces, me enfoqué particularmente en las vías respiratorias y te puedo decir que también es a nivel de qué tipo de células están siendo reclutadas que están causando la inflamación patogénica en estos ratones. Que rápidamente, o sea, neutrófilos, todos esos se vienen a la mente. Porque esto es antes de cualquier inmunidad adaptativa. Esa es la respuesta del IL-6. Sí, excelente. Y ¿qué piensas de la medicina personalizada? La medicina personalizada, hay mucho campo avanzado. Todavía hay, creo que es una pregunta interesante porque creo que la medicina ha avanzado más rápido que la política. Hay muchos estudios ya haciendo a veces de voluntarios que están dispuestos a hacerlo, pero la política, y no solamente la política, la ética, no ha avanzado a paso tan agigantado como lo ha hecho la medicina. En realidad, o sea, yo te puedo decir que ya hay casos, específicamente en Regeneron, donde se ha restaurado la pérdida del oído totalmente de alguien que no escucha bueno, es una enfermedad específica de un gen que está disregulado en el oído, pero totalmente, o sea, ya está ahí. ¿Qué sigue? ¿Cómo lo va a recibir la gente? ¿Cómo lo va a recibir el público en general? Entonces sí creo que ahí es algo que la ética y la política no han avanzado a pasos tan rápido como lo han hecho los terapéuticos.